Due to their indispensable role in genomic DNA replication, DNA helicases have been studied for their potential as chemotherapeutic targets, such that the inhibition of their activity forces cancer cells to undergo mitotic catastrophe and eventually cell death. One of the more recently studied helicases is the minichromosome maintenance proteins 2 to 7 or MCM2 to 7 complex, which is part of a family of ATPases associated with diverse cellular activities or AAA proteins. This heterohexameric protein complex forms the catalytic core of the eukaryotic replicative DNA helicase called CMG complex. More importantly, studies have found that the dysregulation of MCM227 can lead to abnormal cell proliferation and genomic instability, which cellular oncogenes take advantage of. In fact, various cancer tissues and carcinoma cell lines have been found to exhibit an overexpression of MCM proteins. This, and other observations surrounding the roles of MCM proteins in cancer, makes them highly potential for diagnostic, prognostic, and chemotherapeutic purposes. Therefore, this in silico study entitled Ligand-Based Pharmacophore Generation, Virtual Screening, and Molecular Docking Studies of Bioactive Compounds against MCM467 Protein Complex aim to discover novel and potential chemotherapeutic agents from bioactive compounds which inhibit the helicase activity of the MCM467 subcomplex through targeting the interface ATPase sites. To do this, computational methods were utilized. First, the protein and ligands were prepared and minimized using the softwares OpenBabel and UCSF Chimera. Then, a pharmacophore was generated and validated through Ligand Scout based on the 9 known inhibitors of MCM467 from literature. Using this pharmacophore model, 40,500 bioactive compounds from Chembl and NPAS databases were screened. The top screening hits were then docked in the interface ATPase sites MCM46 and MCM74 through UCSF Chimera. The top 10 hits for each binding site with the highest binding energies were then collated, visualized, and further evaluated according to their pharmacokinetic properties. Figure 3 shows the 3D and 2D visualizations of the ligand-based pharmacophore model generated from the training compounds. Shown are the features shared or similar between the training set in 3D space, such as aromatic rings, hydrogen bond acceptors and donors, and positively ionizable moieties. After screening 40,500 bioactive compounds against the model, 151 top hits were chosen to be docked in the interface ATP binding sites. Shown on Table 2 are the top 10 hits for each binding site, 3 of which were found to bind to both sites for a total of 17 leads. All of these compounds have binding energies equal to or higher than the binding energy of the ATP analog ATP gamma S, which suggests that they can competitively inhibit ATP binding. As for their pharmacokinetic properties, the compounds unfortunately exhibited poorly, but they are synthetically accessible and are mostly non-carcinogenic. In line with these results, it is recommended that further studies such as lead optimization, QSAR, and structure-based virtual screening be conducted to improve the pharmacokinetic properties of the presented hits and to explore a wider chemical space. In vitro assays can also be performed to assess the inhibitory activity of the leads against MCM227 or other ATP-dependent helicases.